Okay, well, I'd like to welcome everyone to our monthly webinar series, and this is a series by the Human Islet Research Network. I'm sure most of you have joined us before, but if you haven't, welcome. Today, we're going to focus on chips, and we have three terrific speakers that are going to tell us about the state of the art and what's going on with microfluidic chips and how we can use those to understand type 1 diabetes. For those of you that have been here before, you'll know these slides are somewhat familiar. I wanted to share with you a little bit about the network. This is roughly what our network has looked like. And when NIH funded this, this initiative back in 2014, the primary goal, and still is to understand how human beta cells are lost in type 1 diabetes and try to figure out ways to prevent or protect beta cells and essentially develop pioneering strategies to help people with the disease. Now, things have changed because the network doesn't stay stagnant, and this is what the network is looking like. Now, the latest addition to the network, I'll draw your attention down here, is called PANC-based. This is a pretty ambitious initiative. It was just announced just a few days ago, and there are two groups. Kyle Galton's group at the University of California, San Diego is the lead or contact PI there, and Stephen Parker at the University of Michigan. And this group is going to bring all of the HIRN data and all pancreas data around the world, bring it together in a database. But it's going to use all the data that this network and a number of others have generated, but that's the focus of what HIRN is doing. Now, it's not a closed network either. There's an RFA on the streets that I wanted to make you aware of. The deadline for it is March 20th. Some of you may already know about it, but I'll just tell you a little bit more about it briefly. However, over the course of about 10 years now, there have been 800 or so researchers that have come in and out of HIRN. They are all over the world in different countries, and we've got many institutions. So this really represents a pretty good picture of investigators in type 1 diabetes research and bringing a lot of talent and diversity to bear to ask fundamental questions in terms of the disease. So in terms of the funding opportunities, if you want to know more about these, you can subscribe to our newsletter. It's not all about funding opportunities. It's about job opportunities. It's about the latest developments in the network. And this is the initiative that I was telling you about. You can probably Google that and see what that is all about. But again, that's coming up here in about a little less than in a month. If you want to know more, I'm sure you probably found our website. I'll highlight to you funding, and you can go through and learn more about the various resources, antibodies, data sets, constructs, tissue that's available. Those are all listed for you here, as well as the different databases that might be relevant to you. Also, if you want to follow us on different social media accounts or outlets, you can do that as well. We take a lot of what people are doing in HIRN and we try to catalog that. So if you have, let's say, an antibody that you're interested in or looking for a mice that might suit your experiment, this is a resource that allows you to quickly look through almost 1,000 papers now to try to find what you're looking for. Today's webinar is going to be posted, so you don't have to frantically take notes or screenshots, but we do have a YouTube page here, and so it'll show up within a matter of days there, as well as you can see all of our past webinars. So today, if you would, just make sure that you stay muted during the presentations. We're going to hold questions to the very end. Also, turn off your video as well. That'll help with maybe any bandwidth issues on your side. Again, we've got great topics that we're going to talk about today. And briefly, Ashu, why don't you go ahead and take the screen from me? And while you do that, I'm just going to briefly introduce you and the other speakers that we have in front of us today. So first up is going to be Ashu Agarwal. He's an associate professor in the Department of Biomedical Engineering at the University of Miami, and he's been with HIRN for quite some time, leading some of the efforts in one of the consortiums that I showed you, CHIB. Dan Hui is an associate professor in the Department of Bioengineering at the University of Pennsylvania, and Dan has some of the coolest images that I've ever seen on screen. Chris Hughes is a professor in the Department of Molecular Biology and Biochemistry locally here at UCR. So these guys have a wonderful talk prepared for us, so I'm excited to hear what's here. Thank you, Ashu.
One minor correction. I'm sure Chris wants to jump in.
Uh-oh, what did I get wrong, Chris?
I'm at UC Irvine, not UC Riverside.
Oh, man. You know what? That's where I went to school. Sorry.
All right.
Sorry about that, Chris.
No worries, John.
Thanks,
And thank you.
All right.
Thank you for an awesome introduction into HIRN. But you also forgot to mention the coolest consortium within HIRN is the CHIB consortium.
I knew I was going to get into trouble.
So the three of us represent the CHIB consortium, which is on human islet biomimetics. This is the sort of the alphabet soup of the HIRN consortium, and each of them are serving a very useful purpose in our overall fight against type 1 diabetes. It is, needless to say to this audience, is very exciting times if you're a type 1 diabetician or a researcher, because within the past decade or so, we've gone from experimental clinical islet transplantation all the way to a real commercial entity doing first in human trials, but also now expanding those trials in various phases of clinical, trial phases, and then not just with immunosuppression, but also now new strategies where you would not need, hopefully, immunosuppression. So where do bioengineers fit into this, and where does the CHIB consortium fit into this? So CHIB has evolved. It started just like HIRN in 2014, and we're 10 years later now. And the initial focus was essentially on making sure that the islets are happy. We understand how islets live within their niches, and how we can leverage creating an ideal niche to maintain an islet function, the shelf life of these islets for prolonged periods of time. We transitioned into how not just human-derived islets, but stem cell-derived islets could be matured and interrogated before transplantation and expanded, and how bioengineers, and CHIBs can help in that. The current version or the iteration of the CHIB consortium is on modeling autoimmune, islet-immune interactions and autoimmune pathogenesis, and the RFA that sort of John mentioned expands islet-immune interactions in an isogenic system to also perhaps look for therapeutic targets and disease and drugs. So as John said, there would be three quick vignettes. The first one is from Dan Hui, so I will stop sharing and let him share. The next one would be with Chris, and I would finish it off at the end. So Dan, I shall stop sharing. The floor is yours.
Okay, so let me share my screen here. Okay, can you guys see the whole slide, or are you seeing the presenter view?
Seeing the presenter view.
Okay, so let me. How about now?
Got it. Good.
Okay, all right. So let me go ahead and get started. So my name is Dan Hui, professor of bioengineering. I got promoted last week, and so I also lead a research group. Let me actually. Thank you, Chris. Yep. A research group called BioLines Laboratory. It's acronym for this long name shown here. So I usually start my talk by showing a photo of my group. I have about 14 people working in my lab, half postdocs, half grad students. And we identify ourselves basically as a team of bioengineers working on a technology called human organ on a chip. So I was told by Leila and John that many of the attendees in this webinar might not be familiar with the idea of this technology, or maybe the technologies and approaches that we use. So what I'd like to do is to maybe briefly explain the gist of this technology approach, and then deep dive into maybe some of the models relevant to this HIRN consortium happening in my lab. So what we do in my lab basically revolves around this idea of using microfabricated devices for biomedical applications. And so what's shown here is a plastic chip. It's actually made out of this silicone rubber called the polydimethylsiloxane. In case you're completely new to microtechnology, microfabrication. So what we do to fabricate these devices is we actually use fabrication techniques initially developed for manufacturing these computer chips. So this technique, one of the essential techniques that we use is called photolithography. So this refers to a set of techniques that allow you to make small-scale structures using light. So what I'm showing here is a simplified, workflow of photolithography. We typically start with this silicone wafer, and then we deposit a thin layer of a photosensitive polymer. And then we then place this photomask that contains the shape of the feature that you like to generate on the silicone wafer, and expose the whole thing to UV. So during this UV exposure step, the UV sensitive polymer sitting in this transparent area undergoes chemical reaction. And one way to think about it is actually the exposed photosensitive polymer is cured and hardened, whereas the rest of the photoresist remains kind of liquidy. And during the following development step, uncured photoresist dissolves away. And as a result, we can generate this small-scale feature on the surface of a silicone wafer. So this is actually a conventional technique we used to use. Oftentimes, nowadays, we use simply 3D printed wafers and plastic wafers. We fabricate through high resolution 3D printing companies. And then once we get these 3D printed or photolithographically prepared molds or masters, we bring them to the lab and we put it in the dish. We pour free polymer of this silicone rubber, PDMS, to cure it. So we put it in the oven at 60 or 70 degrees for a couple of hours. It cures, it hardens, and you can peel it off of the master. And when you do that, these patterns, raised features on the wafer, are transferred onto this one side of this PDMS slab, which can then be sealed against a flat slab of another substrate to form this enclosed chamber structure. So maybe this is like one of the most simplified versions of this workflow, but basically photolithography or 3D printing combined with soft lithography are the techniques that we use to build these microfabricated devices that I showed you earlier. So what do we do with these devices? What we do is we actually put human cells, many times primary cells, but also transformed cells or stem cells, and we grow them in these devices, microscopic chambers within these devices, but we also use sometimes nano engineering techniques to build complex 3D structures, and also, most importantly, the cells with the type of environment that they would like, that they would feel at home, if you will. And always our end goal is to engineer realistic human tissues that can be used for a variety of applications. Okay? So this is the idea of microphysiological systems or organ chip technology that all three groups are using in this chip consortium. So using this approach, my lab over the last 10 years or so have developed a variety of model systems, some of which are shown here. So there's a lot to talk about here, but for the purpose of this webinar, what I'd like to focus on is to engineer blood vessels and also vascularize human tissues. And this is, I think it provides a technological basis for the chip work, HIRN work that we're conducting in my laboratory. So let me start with the device that we use to build these, engineered tissues. So what I'm showing here is a microfabricated device made out of the same material, PDMS. And it's about the size of a rubber eraser. This is a cutaway view of the device. There are three partially separated compartments, and these compartments or lanes are separated by these thin protruding structures microfabricated within this chamber. So what we can do in this design or in this system is to inject liquid into the middle compartment. And there's a well-known physics phenomenon called capillary pinning effect. So by action of surface tension, this liquid can be trapped only in the middle lane into which this liquid is injected. So using this principle, we inject a mixture of hydrogel precursor solution in a liquid form mixed with endothelial cells and fibroblasts, and we gel it to embed these cells in this 3D scaffold. We fill the side chambers with media to feed the cells in this 3D culture system. We also seed side channels with endothelial cells. So during this 3D culture, the endothelial cells in this hydrogel, in the presence of all the supporting factors produced by the surrounding fibroblasts and also the matrix, they actually assemble themselves into 3D vessels that open to the side channels. So let me actually show you a movie where this process actually happens in this fibrin scaffold and encapsulating these RFP expressing endothelial cells. And over time, as you can tell, this is a sped-up movie. This happens over 7 days or so, but the vessels form, they interconnect, and then they, as a result, form this 3D vascular network, which can be seen in this micrograph as well. And they are very similar to native human vessels, especially capillary-sized vessels. I'm showing a series of images here to demonstrate the similarities. And another interesting thing that happens during culture is the endothelial cells in the side channels, actually, they form angiogenic sprouts into the hydrogel to connect with the 3D vessels in the gel. And this micrograph is basically a zoomed-in view of this anastomosis happening at this interface. And as a result, these vessels in the hydrogel become accessible and perfusible from the side channels. You can imagine putting fluorescent dye in one of these chambers and then apply a pressure gradient, and you can actually perfuse the entire network in a directional manner in the controlled fashion. You can also introduce blood cells and let them flow and also interact with endothelium, as shown in these movies. So like I said, we are actually using this as a platform to build more realistic models of human tissues, vascularized human tissues. And so very quickly, walk you through some of these examples. We can use exactly the same design to embed vascular endothelial cells and stromal cells, but also CD34 positive human hematopoietic stem cells to make a hematopoietic vascular niche in the marrow that can produce red blood cells, white blood cells as shown here. And also this bone marrow chip can be hooked up to infected lung area on a chip. And all the secreted cytokines and chemokines produced by the infected tissue can flow into the marrow to induce massive mobilization of neutrophils that come into the area on a chip to basically recapitulate the entire process of innate immune response and phagocytic activities in this man-made system. We can also put tumor explants or tumor spheroids in these devices. And we're currently using this platform to study how CAR T cells interact with solid tumors. This is one example where we inject CAR T cells designed to recognize antigens expressed by lung cancer cells. And we can visualize the entire process of CAR T cell extravasation and directional migration towards tumors, and then their effector function for prolonged periods. And we can also use similar designs and techniques to vascularize organoids for the purposes of creating vascularized, bigger, functionally more mature organoid tissues as well. And most recently, we have also succeeded in scaling up some of these actually model systems using this platform shown here, containing 256 individually addressable cell culture chambers that can be controlled by very sophisticated fluidic circuit to really scale up the production of tissue and also data acquisition from these model systems. Okay, so before I finish, in this, as part of the HIRN consortium, in collaboration with the investigators at Penn shown here, we're actually creating vascularized human islet tissues. So we used devices shown here, and then we basically use the same design to trap liquid hydrogel containing islets and endothelial cells of fibroblast, and then we gel it. We culture them in this 3D environment to form these vascularized islet tissues. And so we can maintain their phenotype. These are different cell populations for prolonged periods over a month in these systems, as demonstrated in these images of alpha cells and beta cells. When the islets are not vascularized, their longevity is actually reduced, as you can tell by comparison in these images. And you can also retain their functionality like GSIS, functionality as shown in these plots. So to model the type 1 diabetes, actually, or the immune islet interactions, it would be ideal, if you can get islets and T cells from type 1 diabetics, but the logistics of, these kinds of studies are quite challenging. So what we're now trying to do is to use CAR T cells and engineer them to recognize certain, antigens or surface markers expressed by islet cells to kind of simulate islet immune interactions in the system. So just to show you a snapshot, these are the islets we get from donors, and we put them into our devices to vascularize them. And when we put, for example, these CAR T cells are expressing, or actually they're engineered to recognize DPP6 expressed by islet cells, beta cells, and alpha cells. And what I'm showing out here on the left is extravasation of these DPP6, CAR T cells from the vessels and directional migration towards islet tissue here, which exhibits green autofluorescence. And then let me actually, this is the last movie. Yeah, sorry. And then the movie on the right is showing trafficking and accumulation of these DPP6 CAR T cells within the islet tissues in our devices. And so afterwards, we can actually use these images and also device effluence to measure the number of CAR T cells, the extent of CAR T cell trafficking, changes in the volumes of these islets, and also all these cytokines and chemokines produced by the tissues as a result of CAR T cell infiltration. And also we can perform GSIS assays to show altered, the functionality of these islet tissues as a result of immune attack by CAR T cells. So this is actually an ongoing effort, but we're actually, trying to, get more data. And hopefully we'll, submit a manuscript towards the end of, or actually sometime this year, but that's our goal, but you get the idea. So this is actually what's happening in our group as part of the HIRN consortium effort. Thank you.
Thank you, Dan. And if you could stop sharing, there's actually a question in the chat. I think this is a good question for all of us. So Dan, do you want to take it now, or maybe Chris, I'd love for you to also take a shot at it. So maybe let's keep that question towards the end and let Chris present. And then all of us can tackle that, because that's a very good question for all of us. So Chris, take it away. Good. You're on mute, Chris.
I know. All right, can you hear me now?
Yes, sir.
I'm in a Zoom moment there. Sorry, everyone. So thanks, Dan, for going through the basics of microfluidics. That's very helpful. It means I don't have to do it. So what I'm going to do is jump right in, talk about our platform. Those of you who are familiar with it will know some of this already. For those who aren't, works on very similar principles to what Dan showed you. I have to put this up. My university asked me to do it. And that is just to say that the technology that I'm going to talk about is actually being commercialized through a company, and UCI are fine with that. So that's cool. So one of the things that's driving all of us to generate these microfluidic platforms is something called the 3Rs initiative. And this is started in Europe, I believe, and it's spreading over here as well. And the idea is to replace, reduce, refine the use of animals. So we use a lot of mice in our studies. And for many reasons, we would like to move away from using animals and use human tissues in culture as much as we can. And excitingly, the recent FDA Modernization Act explicitly talked about organ chips and microphysiological systems as tools that they like to see and will accept for drug filing applications. So all of us are very excited about the growth of this field. So what we try and do, what all of us really try to do, is capture this sweet spot between very simple systems that many of us are familiar with, just growing cells as a single cell type on a plastic dish. And then at the other end of this spectrum is the very definition of physiologic, which is the human. And mice are okay for some things, not others. There are things called spheroids where you get cells to stick together in little balls. Those are better than monolayers. But what we're all trying to do, as I said, is hit this sweet spot between simple and physiologic. And what you're seeing here is what we call a vascularized microorgan. Similar to what you saw with Dan, at the top, there's a microfluidic artery, at the bottom, a microfluidic vein. In red is a living human vascular network, and we flow a blood substitute through this, just as if it was blood. And then within this vascular network, we can grow any tissues that we want. So it's all based on your vascular system, a high-pressure arterial side, a low-pressure venular side, connected by these capillaries, and it's through capillaries that all drugs and nutrients are delivered to cells in the body. So again, we schematize it this way, with the microfluidics, done exactly the way Dan described it. And over time, these vascular networks will form in this platform and connect the artery and the vein, and that's what it looks like. And all of this can fit on a plastic plate, a 96-well dish, all the fancy microfluidics that Dan showed you. Those are all glued on the bottom of this. This is an industry standard size, so very easy to use. So why are we all going to this effort to put blood vessels into our tissues? The reason all organs are vascularized, vessels help to develop tissues and pattern tissues. Most cells in your body are within 100 microns of a blood vessel. That's less than the width of a human hair. So there's a lot of blood vessels in the body. As I said, all nutrients and most drugs are delivered through the vessels. Immune cells, we're interested in those because those cause damage to islets in type 1 diabetes. Those are delivered through the vasculature. Cells and organs talk to each other through the vasculature. And we do it with human because mouse is not a human. Many scientists apparently can't really tell the difference between a mouse and a human. Anyone who's not a scientist sees some very obvious differences. And so we understand that. So to best model human disease, we want these vascularized and perfused human tissues. And here's just some pretty pictures of the blood vessels. Here we're running beads through, here we're running blood through. This last picture is blood in the skin. And so you can see the blood flow looks very similar in our platform as it does in the skin. So we're here because of diabetes. As you know, there are two types. Type 1 is the hereditary form, usually appears in childhood, and that's very clearly an autoimmune disease. Type 2 is more lifestyle associated. In both cases, high glucose in your blood can damage tissues, and that's one of the problems with diabetes. So we've taken our chip and redesigned it. It's what you saw before, microfluidic arteries and veins connected by these capillary networks. And this light brown is the tissue chamber. So we grow our blood vessels in there, and we're putting in human islets that we collect We also generate islets from iPS cells, so induced pluripotent stem cells. They work very similarly in our hands. So we call this our islet VMO platform. So islets and our vascularized microorgan. And you can see the dimensions of this. These are very small tissues. So this is what it looks like. Everything goes in at the beginning. The blood vessels form around the islets and support the islets. You can see that in the middle image. Here we're running through green blood. So this is like the Hulk, I guess. Our blood happens to be green, and you can see that flowing through these vessels. And the islets also happen to be green in this case. And this is, for those who are interested, this is laminin. That's a protein that is known to be important in this process. So this just shows you, as Dan showed, how these blood vessels interact with the islets. And we've stained on the right for insulin. So that's what these beta cells in the islets make. That's what's in your blood to control the glucose levels in your blood. That's what goes wrong in diabetes. So we're now modeling the interaction of blood vessels and islets. And you can see that in our islet VMO, these islets look wonderful. All three hormones that are made in islets, insulin, glucagon, and somatostatin, are expressed. And you can see relative to native or standard culture that these look great, and they last a lot longer. Standard culture, 2 or 3 days. Ours, we can go out, as Dan said, a couple of weeks at least. The classic thing you want to see is what's called a glucose-stimulated insulin response. So here, the black line shows the increase of glucose in our platform. And then you can see in the blue line, in response to that increase in glucose, the islets release insulin. And then at the end here, we give it a big kick, and they release lots of insulin. So these work as they are supposed to. Interestingly, if we plot this time course against human data, this is patients who've been given breakfast, lunch, dinner, so they get glucose increase in their blood, and in response, they release insulin. And you can see we get a very similar pattern in our platform. We've also done a lot of computer modeling of what goes on here, and this has been quite interesting. What you're seeing at the top in purple and blue are the outline of the islets. These are pictures from real devices. We then imported those into the computer, and we model oxygen delivery. And blue is where the oxygen is being used up by the islets, which are very metabolically active. In D there, you can see the white is the flow of the blood through the vessels, and in red is the delivery of glucose to those tissues. And here in E, in red there, that's the release of insulin from these islets in response to glucose. And you can see the traces at the bottom there, the actual data, the blue dotted line on the left is the glucose increasing in the platform, and then the simulation mirrors that. And then on the right, in response to that glucose, in both the simulation and the real platform, you see this spike in glucose production. So one of the things we looked at was individual islets here and how those behaved. And you can see in this bottom trace that different islets make different amounts of insulin. So islet 3, very good. Islet 1, really not making much at all. So there's a lot of heterogeneity there. And one of the things we noticed, and you can see on the right here, that this islet 3, a lot of the insulin is going out into the extravascular space and is not being picked up by a vessel. So we asked the question, how important are the vessels? So what we did, we engineered in a new vessel. So in C on the right, there's no vessel over the top of islet 3. Just in engineering, we put in a vessel, and then we ran the simulation again. And so in E here, you can see that with this extra vessel there, there's now more insulin production because we're delivering more oxygen and nutrients, and there's therefore more insulin released. And on the right, if we take the aggregate of all of these islets, you can see in that little inset there that it makes a difference to how much insulin is produced by the entire system, because more of it is being captured in this one extra vessel over the top of islet 3. So that just shows you why it's important to have vasculature in our platform. It also shows you how important it is in the body to have vessels very closely around the islets. And then finally, because we're interested in immune responses, what we've done is we've pre-activated T cells to recognize islets. And you can see that they damage these islets here. There's little gaps beginning to appear in that green. We then put these T cells into our platform, and as Dan showed, they get out of the vessels, they migrate into the islet, and you can see here all these little green dots are the T cells are going into that islet and they're beginning to destroy it. And here are just the data. I won't go through it, but it just shows that we can get these cells into the islets. And here at the bottom, in our control lymphocytes, where we've blocked their ability to get activated, there's no killing, there's no red dots. But on the right, where we've used these activated T cells, the red dots show that we are killing cells within the islet. So we now have this really nice model for type 1 diabetes. And things we're doing with this now, we're switching over to a specialized blood formulation. We're using stem cells to generate these so we can make what's called a fully autologous system. And now we're beginning to identify immune-mediated environmental drivers of beta cell damage. So basically, we're trying to understand how it is that the immune system damages beta cells leading to type 1 diabetes. So just to summarize, this is a complex model that captures what goes on in the body. Mice are not people, which is why we need these platforms. And we and others believe that having vascularized tissues is critical for proper functioning of these islets in vitro. With that, here's the wonderful people who did the lab. Hugh and Ben just published this paper, which is wonderful. And thanks to all my collaborators and to everyone else. And thank you. Back to you guys.
Thanks, Chris. If you could stop sharing. Do you want to maybe take the question now? Because both of you just showed data on flowing in the PBMC-derived or CAR T cells. That's on the... It's an important one. Chris, do you want to go first?
Happy to take questions. Yeah, the first one, how do you manage the variability? That is a problem with any time you try and do work in the laboratory. Clinical trials, we use hundreds of patients, kind of captures that heterogeneity. What we've mostly tried to do, I think everyone has done this to begin with, is to establish the platforms with well-validated cells so that you can reduce the number of variables. Once you've got a platform that works and you can believe in it, you can then start taking cells from different patients. And we're beginning to do that by using stem cells. We can get blood from people, and from that blood, we can make iPSC stem cells. And from those, we can make all the cells that we want. We can make the beta cells for the islets. We can make the endothelial cells, the stromal cells. So we can now start to generate person-specific platforms. And then you can start to compare those to trying to capture some of the heterogeneity that you see in patients. But it's a great question. It's not trivial, but that's what we're trying to do because ultimately we want to understand how diabetes occurs in each individual patient because it is an autoimmune disease. So initially we've done these autologous platforms, but with the iPSC, that allows us to do truly autologous studies. So that's where many of us are headed now.
Yeah, one fight at a time.
Yes.
Yeah. I mean, just to add, we think similarly, and so I guess the autologous models would be ideal, especially for the purposes of studying islet-immune reactions again. But like I said, we've tried it, and actually, it's not the easiest thing to do, actually. So this is more controlled and more experimentally controlled and tractable system that we're using. based on CAR-T, techniques. And so, the variability, batch to batch or, device to device variability, in my opinion, is inevitable, like as Chris, pointed out. And so, there are some trade secrets that, different labs use. And in our case, and I'm sure other labs do this, but, once we identify kind of good batch of cells or good, lots, then we reach out to the, to the vendors, and we try to get our cells from the same lot, actually, as a way to kind of QC our experiments and devices and tissues. And we definitely see like, allogenic reactions between, immune cells and our endothelial tissues and parenchymal tissues, especially endothelial cells and, immune cells. So, I think Chris actually, showed this nice result of, blocking MHC, but, we, in our lab, we actually try to focus on the differential interactions, like between, just the native, or just the base, control conditions versus manipulations we apply to these tissues. And if there's differences, it's those differences we try to focus on to tell the story.
Yeah. Okay. Olivia, do you mind if we take a question towards the end?
Sure.
Okay. Thanks, Olivia. So I just wanted to present sort of a complementary set of tools to what Chris and Dan presented. So our devices are rather, large clearance, PDMS-free, non-photolithography generated machine devices into plastic. And that really serve very well, organoids that could be up to 1,000 microns or 1 millimeter, because the clearances of our devices are, 1.5 millimeter or so. They can be up to 2 millimeter, which is 2,000 micron. And so in that regime, you lose the laminar flow advantages. So the family of devices in the lab are all created using these geometric features, both the inflow side at the cell culture area, but also at the outflow side, so as to create a pseudo-laminar flow. And we couple these geometric features, just like Dan and Chris, with computer simulations of not just the fluid dynamics within the platform, but the fluid dynamics are coupled to both insulin release based on local glucose concentration and local oxygen concentration, which is also what, as I just said, Chris and Dan do. So we make our devices outside the clean room through machining in inert plastics, such as acrylic. And then these acrylic pieces have to be physically, through a mechanical force, clamped together to force a fluidic seal. And the clamping happens using a mechanical clamp, this blue clamp that you see, that brings these two interlocking pieces that we've machined together. And in between these two interlocking pieces sit custom-made rubber gaskets, which are these green elements, which are the only compressible units in the whole system. And once there's a mechanical pressure, these compressible units will create independent, in this chip, N of 3. Once we do that, before putting any cells in, again, just like all of us on the call, we verify the fluidic paradigm, not just through simulations, but through experimental observations, through techniques called particle imaging velocimetry, where we can flow in fluorescent particles, and using a camera at different planes and at different times, you can actually map out if your velocity profiles are laminar, or in our cases, at least as close to laminar as possible, and then what the shear stresses are. So with that family of devices, along with Sherry Stabler, who should say has been leading the motley crew over the last 10 years, has shown both in her lab and our lab for rodent and human islets, we can track very nicely the same glucose-stimulated insulin response that Chris and Dan showed. And for human islets, a severe reduction in functionality over time. And that reduction in functionality gets solved by local oxygen supply. At the same time, this model. Or a local supply of oxygen if you build in, either through bringing more oxygen through the fluid, but in our case, we have a parallel or an independent source of oxygen just from the floor, from the bottom of the device. So we can, because we machine everything, we can make a throughcut at the bottom and stick on an oxygen permeable membrane on the bottom. And this oxygen permeable membrane, then we would again solve the oxygen permeation or the diffusivity equation verified by inline oxygen sensors. And once we're happy with the, with the flow parameters and the local availability availability of oxygen as predicted by computer simulations. We then go in. Because the chip can be opened and closed, we can cast obviously free islets, but we can also cast biomaterial embedded islets. These biomaterials could be instructive for the immune response that these islets might experience in a human body. Once the islets are casted in your favorite gel, they can be sealed and fluidic connections made through a simple pump and a reservoir, and they can live in an incubator, or they can live on a commercially available glucose-stimulated insulin response assay, BioRep assay, that's been commercialized by the company BioRep, which many of our colleagues on this network use, or they can sit on a microscope so you can do live imaging. So unsurprisingly, an oxygen-permeable chip maintains islet viability and islet function much better than a solid plastic chip, and those results get also reflected if you actually use human islets and do repeat GSIRs on the same human islet over time. Again, unsurprisingly, having a local enrichment of oxygen and perfusion enables extension of functional lifetime. Now, the platform's quite simple to use, and actually encouraged by Olivier's instructions, we've shared this device amongst various labs at HIRN. And one of my favorite story is the use of these devices in Marcela Brisova and Al Power's labs at Vanderbilt, where they've used their pseudo-islet technology to transfect alpha, beta, and delta cells with ion-sensitive channel rhodopsins and then light-sensitive channel rhodopsins, and stimulated each of those hormone release by a different wavelength and collected the concomitant hormone on the outflow side. So that enabled their nice JCI insight story. And then their lab actually published a very beautiful Jove paper, Journal of Visualized Experiments paper last year, where you can actually see the assembly of the device, but also the use of the device. So for the current version of the grant, where we have to study islet-immune interactions, we took the same design philosophy of using PDMS-free plastic devices and actually maturing these barrier function. For example, endothelial cells, you could have epithelial cells in there. And then we found a way of maturing them, obviously on a transfer outside the chip, but then manually cutting those living bilayer cultures and sticking them in a wet plastic adhesion sterile manner into the chip, which have independent apical and basal flow lines, each of which can be connected to a pump which controls the pressure and flow rate independently in your apical side and a basal side. And the outflow could then be, this is a cartoon, it's not a reality yet. The outflow could be used to measure your, through an array of biosensors, the kind of secretome that you would want to sample from. And so again, just like we do with all our chips, we've at least from a computer simulation point of view, satisfied ourselves that there is a linear increase in the flow rate with the pressure, which would mean that there is not a leakage. And the barrier is quite, even after the transfer of this living bilayer onto the chip, the barrier is quite maintained, and there is no decline in functionality. And then verified those flow rates with the experimental measurements. We've not done the islet-immune interaction. The islet-immune interaction work is not ready for prime time, but at least for this work, we could show, just like in the classic lung that Dan popularized, we can even induce an air-liquid interface and exercise an air pressure wave on the top side to flex the transverse membrane and create sort of a breathing lung or a glomerular barrier for filtration of a urine surrogate, a blood surrogate into urine by podocytes, for example. Now, I'm just going to switch gears and introduce a new biospecimen that was popularized by my colleague here at Miami, Juan Bendala Dominguez, who's on the call, I think, where he pioneered long-term culture of pancreatic slices by discovering that you really need local oxygen environments to be maintained in a pancreatic slice culture. So from a few hours to, he took the life to a couple of days, and by just having, again, just like in sort of similar to our devices, a floor that was oxygen permeable. So these pancreatic slices are very, very interesting as a model system, because here you've got the entire tissue, not just the islets, talking and surrounded in the context of the exocrine and the other cell types that might be important. So he and I started talking to each other, and we thought we could just plop those slices into a chip and flow out the harmful sort of digestive enzymes that would chew up the islet in a static culture. And just having continuous perfusion would help us also maintaining their lifetime. So that project turned out to be a longer challenge, and we just published this in Lab on a Chip last month, where it turns out that if you have so many temperature changes in a microfluidic device, you've got air bubbles, and really the only robust way of eliminating air bubbles is to use a degasser. So really take out all the resident the gases that are dissolved in your liquid. Because when your solution rises up from room temperature to 37 degrees Celsius, the gas solubility decreases. And when the solubility decreases, the gas partitions out in the form of air bubbles. So using degassers is the only robust way of really making sure there's no incubation, there's no propagation of air bubbles within the device. But once you degas it, you're also getting rid of the oxygen. So we used an oxygen tank to drive a pressure-driven pump, which would then get, just to confuse you guys, there was pure oxygen in the media, which was then getting degassed to get rid of other gases except oxygen, and then also a debubbler for good measure. And the slices, pancreatic slices, would sit in the microscope for their entirety of multiple days. And you could image each of those slices. Even with perfusion, they wouldn't move because we created anchors, sort of these 3D printed stainless steel anchors that wouldn't interfere with imaging, but would sit on the outside, sort of the periphery of the slices, so that the slices were held down by these anchors. And so you could come in and do high mag imaging. For example, in this case, these were slices not from a human, but from a mouse where insulin positive areas were GFP positive. So the same GFP positive area could be imaged over time. So of course, in Juan's lab with our devices, we are expanding this towards looking at more interesting neogenic events, for example, or indeed islet-immune interactions, perhaps. But the idea is that the platform is now ready for deployment, and it opens up an entirely new bioresource, which are human pancreatic slices for interrogation. And for human slices, we did GSIS and again showed restoration of function over multiple days. So with that, I will close 7 minutes ahead for questions, and I will just say that we've used commercially available clamps and commercially available fluidic architectures, which are sold by independent companies. But through my lab, through academic and commercial collaborations, we've shared our devices with not just Gainesville and Al Powers, but also WashU, Hebrew University of Jerusalem, and UPMC. And this is the crew that does it, and these are the funding sources. So I will stop now sharing, and we can all look at the questions together.
Thank you, Ashu. Thank you, Dan. Thank you, Chris. Every time I listen to you guys, I feel very inadequate. This is really terrific work, and you're showing us what the future is going to look like. Chris, thank you also for answering some of these questions as well. Olivier, you have your hands raised.
Yeah, I have a question mostly for Chris and Dan. So you both mentioned that in your current system, the islet themselves are not vascular. They sort of lay in a bed of some sort of vascular network. And so my question is, what would it take, and what kind of strategies could you use to vascularize the islet itself?
Thanks, Olivier. We have done that. I mentioned it in one of the answers there. So when you isolate cadaveric islets, there are little fragments of vessels, but they don't seem to survive very well. So what we find is that the vessels wrap tightly around the outside of the islet. So in one sense, I think that's okay. Those endothelial cells that are adjacent to the islet don't know whether they're in the islet or just adjacent. So I think those endothelial cells functionally are in the islet. So that's one thing. We can also make pseudo-islets. And when we do that, we can incorporate stroma and endothelial cells. And then those pseudo-islets do get fully perfused with vessels, fully penetrated by vessels.
Yeah, in our case, we've seen both actually. So this is, I think, is one of the good examples of variability that exists, inherently exists, in these, clinically sourced explants and tissue specimens. In some islets, actually, we see blood vessels within those islet aggregates. And when we put them into our device without any vessels, actually, we've seen like the sprouting and outgrowth of these vessels from those explants to the surrounding tissue. In this case, it's just the scaffold, the 3D scaffold. So in which case, if there are vessels around these islets containing vessels inside, we've seen anastomosis happening. But in many other times, we don't really see outgrowth of these vessels, and we just try to actually increase vascular density to get these vessels as close to the islet explants as possible. But as Chris pointed out, actually, like pseudo islets can be made, or maybe in our case, when we get these islets, we've done an experimental procedure where we break them into smaller fragments and then mix them with the endothelial cells and fibroblasts. This is kind of similar to, the method of making pseudo-islets, but we try to kind of retain the native architecture of these islet explants as much as it can, actually, by not breaking them into single cells, but into smaller fragments, which could then be combined together to form or increase vascularity within the islet tissue compartment. I hope that answers the question.
Yeah. Chris, you've been busy on the chat answering questions via text, so that's good.
I tried a few, yes.
If anyone wants to pose any more questions to our speakers, you can raise your hand or go off mute, and Ashu, I think, will catch it.
Okay. Do you want to close it, John?
Yeah. Well, I want to thank you guys again for taking the time to put this data together and showing us the work that you've been doing, which is really beautiful. We appreciate it, and just want to remind everybody that we have these webinars on a monthly basis. We'll have this up in a few days, I think, so that you all can take a look at this. So thank you very much, everyone, and we'll see you soon. Thank you, guys.
Cheers.
Thank you.
Okay.
Thanks, everyone. Bye-bye.